started here. So thanks for coming today. This is the first of our candidate seminars for the plant pathology position. Robyn Roberts is our guest today. She's going to tell us about her work on in tomato diseases. She got her undergraduate degree in biology at Indiana University, and then did a PhD in plant pathology at the University of Wisconsin, and is now a postdoc at the Boyce Thompson Institute, which is at Cornell University. So I think we'll hear about research that's ongoing there. So, all right, take it away. Thank you.
Thank you, John, and thanks to all the members of the search committee who invited me here today. I'm really excited to talk about some research I've been doing in my postdoc, and it's also really great to see so many familiar faces here, and I'm excited to meet so many more people over the next couple days. Can everyone hear me okay? So I first wanted to start off before getting into my research talk about some of my general experiences in plant microbe interactions. So I really have nearly 10 years of experience in plant microbe interactions using different pathosystems as well. So I started, I grew up in a small town in northern Indiana, surrounded by agriculture, and I participated in 4-H for 10 years, so I've always been really surrounded by agriculture. I decided to go to Indiana University to get a bachelor's degree in biology, and working in Roger Innes's lab, I studied the powdery mildew and Arabidopsis system. And this really made me interested in plant pathology. And from there, I decided to go to the University of Wisconsin in Madison to do a PhD in plant pathology. There I studied plant-virus interactions and studied a virus called triticum mosaic virus, which is a virus of wheat, and really focused on viral replication and really the viral side of plant-virus interactions. And while I learned a lot, and I really like studying viruses, what I thought was really missing was the other piece. So where's the plant side? And what can we do with our knowledge about viruses to help improve plants? And so I decided to move to New York to do a postdoc in Greg Martin's lab at the Boyce Thompson Institute, which is an independent nonprofit affiliate of Cornell. And there I've really been studying tomato immunity, which is what I'll talk about primarily today. So I'll only briefly cover some of the work I did in my PhD. So I was studying triticum mosaic virus. So here are some of the symptoms from a plant I inoculated in wheat. I really studied this unique virus. This virus had emerged right before I started my PhD. And what was really interesting about it was, as far as on the replication and translation side, it had this really unique 5' untranslated region, which viruses use to translate their RNA and really thrive and replicate in the host. This 5' UTR had a UTR that was much more similar to animal viruses. And so this paper that I published in 2015 really highlighted what some of those unique elements were that were more similar to animal viruses. Then in my second publication, I really focused on what are the plant vectors that are involved in viral translation. So I would invite you to read either of these papers, or I'm happy to talk about them further. But I will come back to triticum mosaic virus at the end of my talk today. So the rest of my focus will be on my postdoctoral work in Greg Martin's lab. So I've been there about 3 years now. I started in October of 2016. And I've had 3 separate projects. So the first project was really focused on finding new sources of resistance against Pseudomonas syringae race 1 resistance, which I'll talk about in a little bit, and really understand the mechanisms behind any novel resistance that I would find. Our first publication came out in March of this year, for which I'm the first author. And I also wanted to highlight an undergraduate student who's a co-author on a paper who's an undergraduate of mine. My second project is really to better understand pattern-triggered immunity, and so I'll talk a little bit about that, but I have a paper that I'm planning to submit by the end of the year on some of the differences and similarities between two different immune receptors in tomato that both recognize flagellin. And on this paper, I actually have 3 undergraduate students who will be co-authors on that paper. And my final project is characterizing a new protein that's involved in effector-triggered immunity, which I'll talk about in a little bit. And this protein has been shown to be involved in bacterial, viral, fungal, and nematode resistance. And this paper was officially published in November of this year in MPMI. And I was excited to see it was the MPMI editor's pick for November. So it's open access through the end of the month if any of you are interested in reading. And I also have one other undergraduate who's an co-author on this paper. So I really like all of these 3 projects, and it was really hard to pick one, but in the interest of time, I'm going to focus on the first project today. So Pseudomonas syringae pathovar tomato is the causal agent of bacterial speck in tomato. It causes symptoms on leaves, on stems, fruit, and flowers. It is an economically important disease, as there have been a lot of recent outbreaks, especially in New York. There was a major outbreak in 2015 due to a cool, wet weather summer. And there's also a new race that is emerging that can overcome the only source of resistance that we have against Pseudomonas syringae. So not only is it an economically important disease, but it's also a really great model system for studying bacterial pathogenesis. So the gene PTO is the only source of resistance that we have against Pseudomonas syringae. And you can see that it's very effective resistance. The only difference between these 2 plants is that single gene. And these plants were both inoculated with the same strain. So this gene was discovered about 30 years ago by Greg Martin. And it's been working really well, except in the past 5 to 10 years, where a new race has emerged that can overcome this resistance. So before I get into some of the details of my research, I wanted to first introduce the different ways that plants can recognize pathogens. The first form is through pattern-triggered immunity, or PTI. And in this form of immunity, plants have receptors that recognize conserved patterns on microbes. For Pseudomonas syringae, the major pattern that is recognized is the protein flagellin, which is the motility protein of bacteria, and specifically we're looking here at Pseudomonas. There are 2 receptors in tomato that can recognize bacterial flagellin. One is flagellin sensing 2, or FLS2, and the other is flagellin sensing 3, or FLS3. These 2 receptors, even though they both recognize bacterial flagellin, recognize different peptides or epitopes. So FLS2 recognizes flag 22, and FLS3 recognizes flag 228. FLS2 is found in most plant species, but FLS3 is kind of unique because it's only found in the Solanaceae, and it's not found in all the Solanaceae. So it's in tomato, pepper, and potato, but it's not in eggplant or tobacco. Once the plant recognizes flagellin, it can initiate a PTI response, which includes a whole suite of events, including the production of reactive oxygen species, callus deposition, transcriptional reprogramming, but it's really associated with a moderate inhibition of pathogen growth. So it usually accounts for a difference of about a 10-fold reduction in bacterial growth. But what's also important is because it's recognizing conserved patterns on microbes, there is the potential that we could find resistance against multiple pathogens. So in response to these receptors that recognize conserved patterns through PTI, bacteria have evolved effector proteins to block this form of resistance. And in response to that, plants have evolved effector-triggered immunity, which can recognize these specific effectors. So these effector proteins really aid the pathogen in virulence. At least for Pseudomonas syringae, these effectors are injected into the cell through a needle-like apparatus called a tight secretion system. And the 2 major effectors of Pseudomonas are AVR-PTO and AVR-PTOB. The presence of these 2 effectors defines a race 0 strain. AVR PTO and AVR PTO-B can be recognized by PTO, and in conjunction with the ARC protein PRF, initiates an ETI response. And so, again, a whole suite of events occurs in ETI, including activation of the MAP kinase cascade, changes in gene expression, and really the hallmark is this hypersensitive response, or this localized, controlled plant cell death, where only a few cells will die in order to try to kill the pathogen off. Usually it's associated with a really strong inhibition of pathogen growth, so where PTI is typically thought to be about a 10-fold reduction, ETI is usually about 100 to 1,000-fold reduction. Importantly though, this is also usually race-specific. So what do I mean by race-specific? So again, the presence of AVR PTO and AVR PTO-B defines a race 0 strain, but in the past 5 to 10 years, the pathogen has evolved such that it loses these 2 effectors. And so the loss of these 2 effectors is race 1, and these can no longer, because effectors aren't there, PTO and PRF can't recognize it, and so there's no ETI response. And while we know that a lot of these strains have other effectors, like AVR RPT2 or AVR RPS4, which we know in Arabidopsis there are R genes or R proteins that recognize these effectors, this is not the case for tomato. There are currently no known tomato R proteins that recognize other effectors. So our only source is PTO, and with these race 1 strains emerging, we don't have resistance against this pathogen. So that's really the motivation behind my project. So we're trying to identify new sources of resistance, either a strong PTI or an ETI, and integrate this into breeding lines. We also want to understand the molecular mechanisms behind resistance and susceptibility of these novel genes. We'd like to look at the possibility that we could have some broad spectrum resistance if we find new PTI candidates. And finally, we'd like to identify some susceptibility factors that we could breed out of tomato. So this project has been a big collaborative project that's funded by the NSF and has 7 co-PIs on this grant. And what's really great is that we have experts in plant microbiology. So Alan Colmer and Greg are both at Cornell and BTI. We have 3 people who are bioinformaticists, and I work especially closely with Susie Strickler. And then we have 2 tomato breeders who I also work really closely with. We also have a number of collaborators who run a lot of our field trials and provide other materials for us. So overall, our goals of this project are to screen about 200 accessions for new sources of resistance, identify the genes and develop markers for breeding programs. Simultaneously, this grant also has a large CRISPR project where we're trying to generate 150 different CRISPR lines in immunity-associated genes. And this project is led by a postdoc in our lab, Ning Zhang, and together with Zhangjun Fei and Yi Zheng, who is a postdoc in the Fei lab, they've created this plant CRISPR database. And so I'd invite you to go check it out, plantcrispr.org. It's basically this big database where you can put in any plant species that you're trying to generate a CRISPR line for. So one that you can see who else is already generating the lines and possibly request seeds from them. Anything that we would identify from the screen would go into our CRISPR pipeline. And anything from the CRISPR or the screen, we would integrate into breeding lines with the help of our breeders. And of course, we'd also be investigating the molecular mechanisms behind this function. So today I'm really going to be focusing on the screen. So we know that tomato is naturally diverse, and you can see this if you go to the supermarket or farmer's market and you see tomatoes of every different color and shape and size. So Susie Strickler, who's a bioinformaticist on our project, took all of the available sequence information for tomato, which is over 1,400 accessions, and generated a neighbor joining tree to represent all of tomato diversity. This tree has 83 clusters, and from these 83 clusters, we chose 216 accessions to represent all of the diversity. So the strategy here was to reduce the number of plants, but have a better representation of all tomato diversity. Accessions were chosen based on the size of the cluster, so if it was a larger cluster, we tried to choose more accessions, but it was also somewhat limited on seed availability. The accessions included heirlooms and land races, breeding lines from the 4 major tomato breeding programs. It included the cerasiforme, which are the cherry tomatoes. It included processing and fresh market. And we included 1 wild species, Solanum pinnellifolium, which is the most closely related wild species of tomato and is thought to be the progenitor. Our seeds came from all over the world, including Israel, France, Spain, and the United States. We also ended up using 3 different strains in our screen so that we could easily distinguish between ETI and PTI in the first round of our screening and really speed up our screening process. I won't talk too much about how we chose our strains, but basically by comparing these 2 strains, we could deduce an ETI-like phenotype. And importantly, we chose to use a New York strain that was isolated from the 2015 outbreak in hopes of finding resistance against a New York-specific strain. By comparing these 2 strains, we could deduce a PTI-like phenotype, specifically assaying for flagellin recognition. So to conduct this screen, we planted seedlings in these trays that hold 54 plants. And then Greg and I built this box for about $20 to hold 8 different trays. So we could screen a lot of plants at a time. We spray inoculated to mimic a natural infection, which actually turned out to be really important, and hopefully you'll see why in a minute. And in this box, the whole point was to maintain 100% humidity to aid the pathogen infection. We then scored the plants for response at 3 and 6 days post-inoculation. And I'll mention that a technician in the lab, Sammy Maniero, did most of the inoculations, and my undergraduate student, Alex, did most of the sowing, which was a lot of sowing. We rated the plants on a 1 to 5 disease rating scale, where a 1 had no specs, a 2 had a few symptoms but was overall pretty resistant, a 3 had moderate disease, a 4 was pretty sick, and a 5 was basically dead. Looking at the distribution of scores on our 1 to 5 disease rating scale, we saw a pretty wide distribution depending on the strain that was involved, but overall we're really focused on anything that looks pretty resistant, so less than a 2, and everything that's pretty susceptible or greater than a 4. Importantly, we didn't find any tomato accessions with an ETI-like phenotype. And this actually goes along with what's been found so far in the literature. So there have been other screens, especially in the early 80s and 90s, to look for more sources of resistance. Granted, they usually used only a few accessions and limited strains. So I think this is a broader screen to really show that there is no ETI in tomato. And importantly, PTO and PRF are actually not from tomato. They come from a pinnellifolium integration. So if we want to find a new ETI-like phenotype, I think wild species might be additional sources. And we actually have an example of this from our own lab, where a really fantastic graduate student, Carolina Molina, has been working on this locus called PTR1 that she discovered in Solanum persicoides, which is another wild species of tomato. And it confers resistance against race 1 by recognizing AVR RPT2 and also RIT-BN from Ralstonia. So it confers resistance against 2 different pathogens. And for anyone who knows the Arabidopsis literature, it does not work the same way as it does in Arabidopsis, and the sequence is not at all similar. So hopefully she'll be submitting that paper early next year, so you'll get to hear the rest of the story later. So even though we didn't find any ETI responses, we did see a lot of natural variation from PTI. And so out of all the accessions we tested, we actually found a large number, so almost 10%, or 20 accessions that had this really strong differential response to flagellin. So here is an example. So this is our regular lab strain DC3000, which naturally has flagellin, which is encoded by this fly seed gene. This strain has been engineered to have flagellin deleted by deleting our fly seed gene. And so you could hopefully see here that while there are some symptoms on this leaf, it's a lot worse when we delete flagellin. So flagellin is definitely being recognized by this accession. So to look at how flagellin might be getting recognized in leaves, we measured the bacterial populations in leaves of plants that had been spray inoculated with strains that either had flagellin or did not have flagellin. So here in these bars, I put a plus sign if the strain does have flagellin, and then strains with minus sign will have a minus. So we spray inoculated these and found that by day 2, they had grown to about a log 4, but the strain without flagellin grew 150-fold greater compared to the strain with flagellin. And this was really surprising to us because all the other literature suggests that PTI only accounts for a 10-fold difference. And here in our screen, we're finding accessions that have a much, much stronger PTI phenotype. So then we wanted to know, since we're spray inoculating these to mimic a natural infection, can we also see this if we vacuum inoculate, which would look at recognition in the apoplast. And so my undergraduate student Alex did this vacuum inoculation and found that yes, flagellin is recognized in the apoplast, but at only about a 10-fold difference. So that really follows the literature suggesting that maybe there really are a lot of accessions out there that recognize PTI really well, but we didn't maybe use a naturalistic infection to do that. So I think the inoculation method is important, and also it just shows that there's a lot of natural variation in tomato that we can leverage for resistance. So while we did account for ETI and PTI, what we didn't expect was to see a number of unusual symptoms. So we saw a lot of kind of typical spec symptoms that have these necrotic spots and these yellow halos, which are caused by a hormone mimic in bacteria called carnitine. But we saw a lot of unusual symptoms that are not typically associated with spec. So we saw stem lesions, we saw these shot holes where the lesions dry up and fall out of the leaves, chlorosis, water soaking, necrosis, severe abscission, necrotic spots, and one that I'm really interested in following up are these stem galls. So all of these phenotypes are really interesting to us. We still don't know if it's really a resistance phenotype or a susceptibility phenotype, but for now we're focusing on the stem galls because it's a really, I think, a cool phenotype, and it's also really easy to score. So this is what it looks like if you take a cross-section. So hopefully you can see that there's a strong delineation between the stem tissue and the scald tissue. And we also know that these galls will only form if we spray inoculate the plants in the high humidity. If we use a vacuum inoculation or we don't include the humidity, we don't get these symptoms. They mainly form on the wild species Pimpinellifolium, but not all of them. So about half of them that we inoculated had galls and half did not. There also seems to be a strain-specific requirement. So with our lab strain DC3000, we saw the galls, but with the New York strain PS205, we didn't really see the galls. When we isolate the galls and sort of sterilize them, we can get living bacteria out of them, suggesting the bacteria are really living in these galls. And finally, we know that in an F2 population, we see a 3-to-1 segregation of galls versus no galls that is recessive. So we're currently doing some bulk segregant analysis to map the associated loci. So to summarize this first part of the talk, we really saw a broad range of host responses to Pseudomonas, including some previously unseen symptoms. We know that PTI plays a large role in defense at the leaf surface, especially but also in the apoplast. And we didn't find any evidence of ETI related genes. So where we are now is really trying to find what is the genetic basis to these observed phenotypes. And we're still pretty early in the process, but this is basically where we are in the project, is to identify the genes and develop markers for breeding programs. And how we're doing this is by identifying segregating F2 populations, screening the F2 individuals and pooling them based on resistance and susceptibility, doing the Illumina whole genome sequencing of the pools, and mapping by sequencing and fine mapping to identify the genes. And the screening is being done by Sammy Maniero, and the mapping by sequencing analysis is being done by postdoc Adrian Powell. So I'll briefly talk about kind of where we are in this first part of the segregating F2 populations. So our strategy is to screen, so far we've probably screened about 40 different populations with all 3 of our different strains. And right now we're really focused on things that appear to be simply inherited. So I recognize there's probably a lot of potential for QTLs. But because we have so many candidates, we're focusing on the easiest ones first. So we're looking for 3-to-1 or 1-to-2-to-1 ratios, which hopefully will also be easier for breeders to get into their breeding lines. We're using parents that are already of commercial interest to breeders. So Sam Hutton and Delete Panthy are generating all the F2s and crossing them already into lines that they're using in their breeding programs. So hopefully it'll be really quick to get it introduced, any new resistance. And Sammy is doing the F2 screening. And from all of our populations, right now we're really focused on about 5 F2 populations. So here's an example of 3 different populations that appear to be segregating simply for race 1 resistance to the New York strain. So I think this is really promising. Hopefully you can see that we're seeing a clear difference between resistant and susceptibility. And I think the ratios look really good, so they look like they're a 3-to-1 ratio. And so right now we're really in this process, which is generating the Illumina reads and doing the mapping by sequencing. So to summarize this part, we're really focused on about 5 F2s that seem to be simply segregating for resistance. We're also really interested in mapping that Gall phenotype, and we actually recently generated a de novo Pimpinel folium genome using nanopore sequencing. So right now we're in the process of doing all the annotations. And we also plan to test additional F2 populations that our breeders are currently developing that we haven't yet been able to screen. So for this last part of my research talk, I'd like to focus on kind of the application of where we're going with all of this. So basically we're wondering, can we use Pseudomonas as a probe to find new sources of PTI-based resistance? So from our screen, we identified about 20 different accessions with enhanced PTI to Pseudomonas. And so we teamed up with 5 other collaborators to test them in field trials all across the eastern United States and really look for broad spectrum resistance. So each researcher tested different bacterial pathogens. So in Ithaca, we tested bacterial speck. Chris Smart in Geneva, which is just a little bit north and west of Ithaca, tested bacterial spot, which is caused by Xanthomonas, and bacterial canker, which is caused by flash factor. Ben McGrath tested bacterial speck on Long Island because it is a real problem and growers really want to be involved. In Ohio, Dave Francis tested 3 different species of bacterial spot. Delete Panthy tested bacterial speck in North Carolina, and Sam Hutton tested both bacterial spot and bacterial wilt, which is caused by Ralstonia. So here are some pictures of us transplanting. I have a great lab that helped me transplant all my 600 tomato seedlings by hand on a typical lovely Ithaca June morning. And this is us backpack spraying the inoculum onto the plants about a month later. And here's Greg out here helping us as well. So he's really involved in the field as well. Our inoculations worked really well. So we saw a clear difference between susceptible and resistant lines and some really nice symptoms. So overall, out of the 20 accessions we put out there, 11 of them appeared to have broad spectrum resistance against multiple bacterial pathogens, which is really exciting. And 2 of them actually, that were supposed to be controls for Pseudomonas, turned out to be resistant to other things, which was a little bit of a surprise, but it could be promising. And for example, so the red R's are resistant and the green S's are susceptible, and this yellow MR is moderate resistance. And for example, this accession number 7 was resistant to almost everything we put on it. So this is really promising for us. We've repeated it this year, but we're still analyzing the data. So the data looks really promising so far. So we're pretty excited about this project. So to summarize, we found of the 20 accessions we put out there to test broad spectrum resistance, 11 of them showed resistance to multiple bacterial pathogens, and we're still analyzing the 2019 data. And so our future directions are to map the genes segregating in F2 populations, develop the CRISPR knockouts and any interesting gene candidates, characterize the gene candidates, and also follow up with these broad spectrum resistance in the greenhouse to really confirm the phenotype and hopefully map the loci. So I wanted to talk a little bit about some of my plans for the wheat pathology program here at Colorado State. So my research program will really be motivated to find solutions to high threat pathogens of wheat using basic research. And I think the high threat pathogen is really important so we can really help our Colorado growers. And really I'd like to focus on understanding the biology, genetics, and molecular mechanisms of plant microreactions, better understand some of the fundamental mechanisms of resistance in wheat, and discover transferable traits and solutions to disease problems. One particular pathogen I would be interested in, or disease I would be interested in, is bacterial leaf streak, which causes these symptoms on leaves, and they can also cause this purpling on wheat heads and blooms. And this is caused by the bacterial pathogen Xanthomos translucens pathophyrulosa, or XTU. And there's currently no resistance or chemical control. And as far as I understand, it's really been a growing problem in Colorado. And some research studies show it can have up to a 40% yield loss in severe cases. And it really likes humid, warm environments. So there's the potential that with climate change, it could become increasingly problematic. So my efforts toward resistance against BLS would be to screen wild species and accessions and breeding lines for resistance. So some examples could include emmer, goatgrass, or triticum species. I'd also really like to use multiple strains, similar to what we did with our tomato screen, and use both naturally occurring ones that are important for Colorado, and also ones that we've engineered so that we can do some of the analysis like we did with tomato and really see a difference between ETI and PTI in the first round of screening. I'd also like to look at some potential susceptibility genes that could be linked to these transcription activator-like effectors or TAL effectors, and also collaborate with breeders closely to really make sure that there's a pipeline to introduce novel resistance. I'd really like to study the molecular mechanisms behind resistance genes or susceptibility loci. And I think it's also really important to capture pathogen diversity in fields. So survey and sequence, but also I think characterization is really important. So there are a lot of sequences available for XTU for various strains, but very few of them have actually been characterized. So I think that's a really important step. And I'd also like to use CRISPR-Cas9 as a tool to study resistance and susceptibility. A second pathosystem I'd really be interested in are some viral diseases of wheat. So as I mentioned in my PhD, I studied triticum mosaic virus and really focusing on how the virus replicates. And so I would be interested in studying several viruses, including Trichome mosaic virus and wheat streak mosaic virus, which are both vectored by the wheat chrome mite, as well as potentially some other viruses like barley yellow dwarf that's vectored by aphids. All three of these viruses are some of the most important viral diseases of wheat. I know there are a lot of viral diseases of wheat, so I'd be interested in talking to all of you about where you think the efforts should really be focused. But I do know of those three viruses, right now the only form of resistance is a quantitative trait against wheat streak mosaic, and you can't control the wheat chrome mite with chemicals, so it's a problem. So my efforts toward resistance against viral diseases would be to use gene editing tools to study these interactions. So potentially generate CRISPR-Cas9 knockouts of different translation factors which are known to be involved in viral replication, or some other proteins that are known to be involved in viral replication. I'd also like to look at virus populations, so if we do find something that's resistant, doing some serial passage to see how long it takes to overcome the resistance, so we can know if it's worthwhile putting something like that out in the field. And also, just, really, I think it's important to look at these virus interactions with multiple viruses. So instead of just looking at Trichome mosaic virus or just looking at wheat streak mosaic virus, since these are both transmitted by the same vector, they're often found synergistically. And so maybe some of these interactions change when multiple viruses are present. And finally, I'd like to look at viral field isolates to really capture the pathogen diversity and population changes. So I've been pretty involved in some visioning at BTI recently, including for the Institute for their decadal vision and for our postgraduate society. I'm the co-chair, so we're doing a lot of visioning right now. And so I thought I would kind of put out my vision for the wheat pathology program at CSU. So my 10-year vision would be that Colorado wheat would be resistant to major bacterial and viral pathogens, and we'll have a better understanding of how resistance and susceptibility works in wheat. My mission would be to conduct screens of wheat and its relatives to find new sources of resistance, use genetic modification as a tool, and survey and sequence pathogen isolates. My 5-year goal would be to discover new forms of resistance against the pathogens and better understand the wheat immune system. And then hopefully by 10 years we can bring some of these new forms of resistance to the growers to improve wheat. And my values in my research program, some of them would be innovation, sustainability, grower interest, translational science, collaboration, ethics, and diversity and inclusion. So to kind of summarize, because pathologists love triangles, I thought I would represent this in a triangle. So I'd like to discover and characterize resistance genes, understand the molecular biology and plant microbe interactions, and survey and sequence pathogens for foods. So with that, I'd like to acknowledge my lab, so especially Greg Martin, who's just been a wonderful postdoctoral advisor, Sammy Maniero, who's the technician who helps me a lot with the screening for this project, 2 of my undergraduates, Alex and Ling Wei, who are just absolutely phenomenal. They're doing experiments for me as I speak. They're just really great. They're both applying for graduate school. I've actually had quite a number of undergraduates at BTI, and all of them have participated to some extent in this project, so I'd really like to acknowledge all of them. Also our collaborators, so Adrian and Susie, who've been really our bioinformaticists and really involved in this project, but also all of our other collaborators who have been really involved in our field trials and providing other materials. Our wonderful greenhouse staff who water my plants for me every single day. I know I'm spoiled. And also our funding comes from the NSF for this project, BARD for my FLS3 project, and my contact information is on the slide, so feel free to email me if you'd like. Otherwise, I'm happy to take questions.
It's a FLS2 3 question. So, artifacts. growing out of nature, it's encountering a lot of flagella, right? So how do you want me to go from traditionally the molecular mechanism that's taking part in the greenhouse or growth chamber to where you're seeing flagella constantly? And
Yeah, so your question is basically, so FLS2 and FLS3 both recognize flagellin. What does that mean out in nature? And, you know, we still don't really know why FLS3 is there, because FLS2 recognizes flagellin just as well as FLS3. And that's one of the projects I've been working on. We generated some CRISPR-Cas9 knockouts in FLS2 and FLS3 individually as well as together. And it's shown that FLS2 and FLS3 actually equally contribute to flagellin recognition. And when you knock both of them out, no flagellin is recognized anymore. So it's hard to say why, you know, if one is sufficient, why do they have both? Because it doesn't really seem like an additive effect. And so I really wonder if, you know, we don't really know the answer to that yet, but I really wonder if FLS3 had evolved to recognize some other pathogen, and we just don't know what that is yet. Because flagellin is conserved, so if there's another pathogen that has flag 228, maybe that's what's getting recognized. Does that answer your question?
I guess I'm thinking more along the microbiome type question. So if you have a plant with really high levels of PTI, how would the microbes like overall look in a work that's not?
How would the microbes look?
So there's so much flagellin out of nature.
Right.
And so they must always be on.
Yeah, so, so again, the question is, if, you know, there's flagellin anywhere, and if then these things must always be on, right? There must be, you know, some mechanism to really recognize that this is a pathogen, and so we know that by putting flag 22, the peptide in by itself, and the receptor by itself, you get activation. So that in alone must be sufficient, but I don't know if there are other mechanisms then that pathogens in the microbiome are using to evade that system, or if there are changes, you know, small changes in those epitopes that they're no longer recognized. If we look at FLS2 in Arabidopsis versus tomato can actually recognize flag 15, which is only 15 residues instead of 22 residues. Arabidopsis cannot. So we know that there are a lot of differences, even in FLS2, between Arabidopsis and tomato. So, you know, maybe some small tweaks like that can explain it. Yeah, that's a really good question. I don't really know the answer to it. Yes?
Tomatoes are notoriously sensitive to an herbicide like 2,4-D. In all of your research with different pathogens, have you seen any symptoms that you think might be similar to or might mimic herbicide drift problem for a grower?
So the question is, tomatoes really sensitive to certain herbicides. Do we see any phenotypes that look really similar to this herbicide resistance or herbicide sensitivity? We haven't tested any herbicides specifically. We didn't see anything from the screen where, you know, I really thought it looked really similar to an herbicide like phenotype. Everything that we saw, even though some of those symptoms were really unusual, they still reminded me of some kind of a pathogen. But it's definitely something that we need to follow up because we still don't know if a lot of these phenotypes are really resistance or susceptibility. I will say that another project that I was working on that I mentioned, this protein that's involved in effector-triggered immunity, we generated a CRISPR-Cas9 knockout of that, and it basically kills the plant. So we didn't get any homozygous mutants of this gene called MY1. When we have a heterozygous plant, it's this shrimpy little thing compared to an azygous or wild type. And it's so sensitive that if you basically try to even mock inoculate it, it dies. And in the greenhouse, they sprayed for, I think, thrips. I don't remember the exact pesticide, but it completely killed the plant. So I think that there are some out there that are, I think we also think MY1 is related to development, so that might also be why. But But yeah, that's also a really good question.
Sort of along those lines with thinking about your work to try figure out the genetic basis of the tomato, of the differences in resistance, susceptibility, and then the CRISPR stuff. Do you think, or from maybe other genes that have already been cloned, that loss of function, at least with, it's easy to not put gene out with CRISPR, but if you want to change the promoter or the amino acid sequence, that's sort of still not quite ready. Do you think that the mutations will easily be mimicked by the CRISPR system?
So the question is, when we're thinking about trying to actually put this resistance out, do we think CRISPR is going to work? So I think it's going to depend, because it's possible we can find completely new genes. Or actually we have an example right now of a gene that we're following up for another project where we know that there's a gene that works in wild tomato, but it's like a wild species of tomato, but in regular tomato, there's an early stop codon and a couple other SNPs, and that's the only difference between regular tomato and this wild tomato that would encode this really great resistance that we're following up. And so I think with the advent of things like CRISPR prime, if you've heard of that, it's a way that we can, an easy way we could potentially fix mutations or SNPs in genes in plants. Well, right now it's only been done in a human system, but hopefully that can be translated to a plant system, and if that works, that could be really translatable. If we're looking at just simple knockouts, I think that's pretty easy with CRISPR. We've been really successful with that. So I think it's just going to depend on what is the gene, what mutations are going to be involved. If it's a completely new gene, maybe there's some other source that we can breed it in. So I think that's a really good question to think about with CRISPR. Definitely for tomato, we're not at a point where CRISPR is going to be accepted in tomato. So that's also something that we're going to have to work on. So I think there are a lot of problems with trying to make CRISPR work in tomato that we can't necessarily solve ourselves. But with that being said, we're really using CRISPR as a tool to see how these things function. And I think that's a really important step to the applied side of biology.
Yes.
So, nice talk. Thank you. So tomato has like a ton of resources. Have you thought about how you're going to deal with wheat, which is a little bit more complicated with less resources?
Yeah, so I think one advantage with wheat is there are a lot of breeding resources. So I know there's a lot of recombinant inbred lines or rills. There's a magic population. So I think I'm going to really leverage some of the skills of the breeders to integrate some of these new sources of resistance. I'd also really like to, with some of these other related species, work in something that's maybe a little bit easier to start, like a diploid plant like barley or something. And then once you can find something in barley, maybe you can go search for it in wheat and see how you can manipulate it or study it better in wheat. So I think a lot of kind of these model systems translating into wheat will be really important. But really, I hope to be leveraging a lot of kind of the genetics resources that are already available in wheat.
So I was wondering, so in your screen, the PTI screen in tomato, so you basically now at the effort of getting the F2s and mapping your genes. So what is the timeline that you have for tomato from screening, getting the populations, being able to have a gene identified or even a CRISPR-Cas, mutated genotype, and how does this compare to what you're planning to do for wheat?
Sure. So the question is, what is the timeline from screening to final product in tomato, and how does that compare to wheat? So the screen took about a year because we also did replicates and a couple other things to really be confident in our results. And then to generate a CRISPR-Cas9 knockout, we can do that to a T0 line, so the initial line, within a few months. and then generating like a T1 line, maybe like 9 months or so. So I think that pipeline is pretty fast for tomato. Finding the gene candidates in an F2 population, it's really going to depend on how simple it is to identify the gene. So if they're in, a lot of repeat regions or something like that, or if it's a completely new gene that's not available or not in our reference sequence, which is Heinz, then it might be a little bit more difficult. But we do have a new tomato pangenome, which encompasses, I think, like 700 different tomato lines, and so they already have this whole pool of potentially new genes, so we can use that pangenome as a resource to see if any of our genes could be pangenome. We've also been doing a lot of resequencing, so using Illumina and some nanopore sequencing of anything we're interested in. That timeline, it probably takes a couple days or a week to make the library and send it off, and we usually get the library back in a month, so that's pretty fast. So I think overall, to get a gene, I think we're getting pretty close to a couple of them, and we started the project about 2.5 years ago, so fingers crossed it'll be about 3 years till we can identify the gene and do some of that characterization. And hopefully, because they're already in F2 populations that our growers are interested in, they can work their magic and get it in pretty quickly within 5 to 10 years. So I would hope that the timeline could be similar for wheat. I know that the genetics are more complicated in wheat since it's not a diploid, it's a hexaploid. So I do recognize that would be a little bit more challenging, but I really hope that the breeders can help guide me along in how to work out some of the genetics. And I know they're going to be much bigger experts than I am in wheat. So I do recognize that I will have I'll have a lot to learn about wheat, but I know I can do it. I have lots of experience in plant microinteractions and different pathosystems, so yeah, I'm hoping the timeline will be similar.
All right. Let's say there's no further, okay.
So going back to FLS2 and FLS3,
Sure.
is there anything known about how they're distributed in plants? Like, are FLS2 on the epidermal surface or only the apoplast or something like that?
So all of these receptors are transmembrane, so they're recognizing pathogens on the surface of the plant. Does that answer your question?
I was wondering, maybe FLS2 is expressed in the epidermal cells on the outside, and FLS3 maybe only in the apoplast or something, and that way it doesn't need to be turned on all the time.
Sure. So the question is our FLS3 equally distributed throughout all cell types?
Yeah.
Essentially. We don't know that yet, so we haven't done any tissue-specific RNA-seq or anything like that, or expression studies to really look at specific cell type comparisons. But I haven't noticed, this is kind of anecdotal, I guess, but I haven't noticed any big differences, when I'm taking samples on like where I'm getting them from the plant. So pretty much any leaf will respond the same to FLS2 or to 522 or to 28, which doesn't exactly answer your question, but yeah, we don't really know